in this network, which is made of 500 granule cells and 75 basket cells, we are now going to look at the impact of different heterogeneities. First start with local heterogeneities and then move on to afferent heterogeneity. The function that we are going to be looking at is what is referred to as decorrelation. The question is, you have a certain set of inputs coming onto this particular structure. We are going to ask whether the output of this particular network is much more decorrelated, or the correlation between the outputs is it lesser than the inputs that it is getting. Right? So, that is the question that we are going to ask, because the dentate gyrus has been considered as a pattern separation network. We will come to pattern separation in a minute. So, this is with reference to a network that is not receiving any afferent heterogeneities. There is exactly the same inputs coming onto this particular structure. And what is plotted over here is the correlation coefficient of the output. This is the histogram associated with that. And if you have a homogeneous population, which means that each and every neuron is exactly the same, and there is only synaptic heterogeneity, nothing else, then you see that the correlation coefficients close to 1 over here, which means that the output is fairly correlated even when the inputs are correlated. But as you start introducing intrinsic heterogeneity, either with synaptic heterogeneity or with fixed synaptic weights, you start seeing a certain amount of decorrelation over here. So, that is one the first level of analysis, where you say that just the introduction of intrinsic heterogeneities introduces a certain amount of decorrelation in the outputs, even if the inputs are exactly the same. This is something which has been known for a long period of time. Nathan Urban, Padmanabhan and others have looked at this basically. And now what you do is you add an additional layer of heterogeneity over here, where you say that because this is the dentate gyrus granule cells, they have adult neurogenesis, and therefore you have this structural heterogeneity, which is referred to as age heterogeneity over here. Right? So, you have immature cells, mature cells and so on and so forth. So, you add that additional layer of heterogeneity, you get, even for the same exact inputs, you start getting a higher amount of decorrelation over here. This is exactly the same plot as this, right? This is a scenario where the inputs are exactly the same. There is no afferent heterogeneities. But when you do have afferent heterogeneities, what happens is that the local heterogeneities don't play as much of a role. They contribute almost nothing over here. It's the afferent heterogeneities that contribute majorly to this, right, to the decorrelation that you observe. So, then Poonam asked, if that is indeed the case, what are these local heterogeneities doing? Are they meaningless? Do we just get afferent heterogeneities and leave it at that? Is that the way we deal with it? It turned out when she subjected these networks to perturbations, I'll switch to that side. So, when she subjected these networks to perturbations where she deleted individual ion channels, that is the advantage of having a conductance based network, where you can delete specific ion channels and ask questions about how exactly the network function changes. So, this is a scenario where there were no structural heterogeneities, and you see that the amount of variability that you observe in the correlation, the output correlation that you are observing. 0 would mean that there is no change in function, and anything away from 0 means that there is a large change in the function. Now, if you introduce structural heterogeneities in this particular network, you observe that the amount of change that you observe, even with the deletion of individual ion channels, is much lesser than what you got over here, and therefore, the system is much more resilient to perturbation when you have these heterogeneities in this particular structure. So, this is with reference to simple channel decorrelation. So, then Sarang wanted to look at pattern separation. So, here you have a set of two patterns which are coming with a certain input correlation, and you feed that to this particular network. So, this is the input to the network. You feed that to this particular network and you compute the output, calculate the output correlation and ask if the output correlation is lesser than the input correlation. So, that is the scenario where the input correlation and the output correlation are exactly the same, and on this side of it, where the output correlation is lesser than the input correlation, would be what is referred to as pattern separation.
And all these models, neuron models, are same in across different blocks.
We'll come to that. Right. So in this case, what Sarang did, this is a much larger network. and it mimics the circuitry of the DG, the dentate gyrus itself. So you have 3,600 granule cells, 180 mossy cells, 500 basket cells, and 50 hip cells, and that's the connectivity that is known in terms of what is there in the DG circuit. And you have excitatory and inhibitory connections. So these are inhibitory cells, and the others are excitatory cells. So therefore, there are excitatory and inhibitory connections that are present. Right? So here the function is the input is being presented, and you are computing the output, plotting output correlation as a function of input correlation for different kinds of patterns, and asking questions about whether this network is capable of performing pattern separation or not. So what Sarang did was, for getting the heterogeneities in individual cell types, he did a parametric search, right? And for each of these cell types, we know the physiological properties associated with them, right? So the granule cell physiology is very different from basket cells, and the mossy cells are different from hip cells, and each of them is different from each other, and therefore we have a set of physiological measurements for each and every cell type, and therefore he did a random search on the model parameters, right? So 10 different parameters, generated 100,000 models of granule cells, and asked how many of those granule cells would match all the objectives for granule cells, and he found a very small percentage of them, 6,378, and so on and so forth. So he had a bunch of valid granule cells, valid basket cells, and so on and so forth, each one of which was heterogeneous from each other. I mean, the parameters were very distinct, even though their functional outcomes were exactly the same, right? So they matched with the functional properties of granule cells, but the parametric combinations were different because it was a stochastic search spanning the 10-dimensional space that was there, right? So we have in the paper there is all that analysis that is out there in terms of how heterogeneous they are and so on.
Excuse me, are these neuron models considering dendritic arborization?
These are again single compartmental models, because I mean, if you have dendritic arborization, you will not be able to have that kind of numbers. It becomes, you can have it, it's just that
Curious, because a lot of people study dendritic computing in these areas, right? So
they do, but I mean, as I said, in our questions were with reference to the network. We do have dendritic computation models separately, we don't use it here. Alright, so he has now a bunch of granule cells which were heterogeneous in terms of their parametric and their functional properties, and basket cells same, and so on and so forth. Now what he did was, he repeated the same kind of search, not in the neuronal level, but at the network level basically. So he said that, okay, I'm going to fix the set of inputs that is going to come in now, right? And there is this 700 number of inputs that is going to expand to this 3,600. That was David Marr's original idea of how exactly pattern separation occurs. You have this divergence, and that sparsifies the network, and therefore the output correlation is lesser than the input correlation. That was the original idea. So Sarang's question was, okay, I will take exactly the same inputs, present it to this network, compute the output over here, and now change only the local synaptic weights here, right? So not change anything else from here, just the local synaptic weights are going to be different, and he is now going to search across these synaptic weights over here to ask which of these synaptic weight combinations would give him pattern separation basically, right? So just going to feed exactly the same inputs over here, let the network do whatever it does, get the output over here, and ask questions about whether the output correlation is lesser than the input correlation, right, across all the different kinds of patterns that he generates. So doing that, he generated 20,000 random networks, and if Marr's idea of this divergence mediating pattern separation were true, then all 20,000 of them would have been pattern separating networks. But here what he actually found was that there were only 47 of them which gave him this pattern separation. He had specific metrics which we will come to in a minute, and he showed that, I mean, these 47 networks, you can see that the amount of pattern separation that you get over here is fairly comparable But if you look at the kind of synaptic weights, so these are the synaptic weights associated with the connectivity that is shown there. You see that there is wide variability in terms of histogram, telling you that there are different network combinations that can give you exactly the same functional outcome in this kind of a network, basically. Right? So how did you quantify this? So this is what I showed you. So that is input correlation, this is output correlation, that's the 45-degree line. This side of it is pattern separation, that side is pattern completion. He rotated this, got this curve along this axis, computed, fitted a polynomial to it, and had a bunch of metrics from there, computed the area under the curve for the negative part, the area under the curve for the positive part, the peak pattern separation index, and the transition point between pattern completion and pattern separation, and the 47 networks that he had. You see that there is wide variability over there also. All of them had shown, as you saw, efficacious pattern separation, but you observe that there is variability in those metrics as well. Right? So there is network to network variability. This was the most important conclusion from his study. So what he said was, okay, if I have this particular network and it is the local synaptic weights that are distinct from each other, so what I will now do is I am going to delete all the hip cells, delete all the mossy cells, delete all the basket cells, and ask questions about which of these interneurons is contributing maximally to pattern separation. So that's the question he asked. So this is the baseline network, and you see that they show efficacious pattern separation over here. When basket cells were deleted, you see that there is no pattern separation component except for one network over here. Pretty much all of them became pattern completing networks. But in this case, there was complete degradation across all networks pretty much. But if you look at the mossy cells and the hip cells, in certain networks you have pretty much no change over here. In certain others you have a huge change, which is true for even here as well. So there is wide network to network variability in terms of how exactly the network responds to deletion of one cell types. So these kinds of experiments have been done by genetically deleting or genetically up to genetically or chemogenetically silencing specific cell types, and the results are all over the place. So this demonstration that even though in the baseline network the amount of pattern separation is pretty comparable, the impact of deleting specific cell types is widely variable across different networks, basically. So because of the different sets of synaptic weights that was present, you have this kind of an outcome. Right? So more relevant to this talk, again, when he subjected it to perturbations and looked at heterogeneous versus homogeneous networks, he found that the impact of these are the different metrics that we saw over here in terms of pattern separation. So you see that the amount of change that you observe in a heterogeneous network to these perturbations, like additive noise did not do much. But if you look at it in terms of low, medium, high levels of noise over here, the heterogeneous network is much more resilient to changes, to perturbations that are introduced into the synaptic weights, or a combination of the two of them. You see that the heterogeneous network was much more resilient to noise compared to the homogeneous ones. Right? So for this part of it, so neural circuit heterogeneities in this case played a beneficial role in decorrelating networks towards achieving high degrees of decorrelation and enhancing network robustness to perturbations. Going to the second part of it, so this is with reference to continuous attractor network. This is work by Divyansh. So this is with reference to what are selective cells that are present in the hippocampus and in the entorhinal cortex. So what is shown over here is an arena, right? And the black lines are a rat moving around in this particular arena, basically. And you are recording from a single cell in the hippocampus, and the red dots over here are the action potentials that are elicited for that particular cell. Right? In this particular case, what you observe is that whenever the animal comes to this closer to this particular location, you see a higher number of action potential elicited. Now you compute the firing rate and map it onto the arena itself. You see that the maximal firing rate is at this particular location, and as you go away from that particular location, the firing rate reduces. Right? So this was a cell type in the hippocampus known as place cells, which was first discovered by John O'Keefe in 1970s. And if you repeat the same experiment in the entorhinal cortex, the medial entorhinal cortex, here what you observe is that these cells don't fire at a single location, but they fire at multiple locations, but it is not arbitrary. You have a specific structure basically. So it's I mean, triangles or hexagons that are present over here. So it kind of tessellates the entire arena that is out there, and therefore they are called as grid cells basically. So it gives you firing in grid patterned manner. So this was first discovered by the Moser labs, and these talk to each other, the place cells in the hippocampus and the entorhinal cortical cells. There are back and forth connections between these two neurons. Right? So we are going to be focusing on this particular thing, the entorhinal cortical structure. So long back, Elafit and Yoram Burak, they had this paper where they showed that you could have continuous attractor network models, a 2D continuous attractor model, which can model this kind of patterned firing in these neurons basically. Right? So one of the problems over here was that this particular paper used exactly homogeneous repeating units, and they were not very different from each other. So Divyansh's fundamental question was, okay, what would happen if I introduce heterogeneities into these structures that are present? I mean, he also recorded from entorhinal cortical stellate cells and showed that they have a wide variability in terms of their properties and stuff like that. So he did not introduce heterogeneity at 1 degree, he introduced it at different degrees, 5 different degrees of heterogeneity. This is intrinsic heterogeneity, where the time constant of the cell is being varied. So 10 was the number that was used over here. So he kept the mean to be exactly the same, varied the variance, and there was heterogeneities across this particular structure. That is intrinsic. Afferent heterogeneity is where the velocity inputs that are coming onto this particular structure, the weight of it is being changed, again by a smaller amount to a larger amount. And third, he also introduced synaptic jitters. So there was this inhibitory connectivity that is present over here, which is what results in that grid pattern firing. He introduced jitter in various levels, and you can see that the root mean square error is increasing with the degree of heterogeneity. So 3 different kinds of heterogeneity. The structural was kind of implicitly incorporated in the intrinsic itself. Right? So he introduced these kinds of heterogeneities. In this case, what happened was, when he introduced these heterogeneities, with intrinsic heterogeneities, you still see the emergence of grid pattern firing. But either with afferent heterogeneity or with synaptic heterogeneity, low level of heterogeneity was all right, but when you entered into higher levels of heterogeneities, the emergence of grid pattern activity was completely abolished. So there was no grid pattern activity, and when you had all of them together, all the heterogeneities together, there also you observe that it is gone. Right? So then he went into the mechanistic understanding of why exactly this is happening, and he found that the difference between a homogeneous network and different heterogeneous networks, the activity differences were predominantly in the low frequency end. Right? So if you look at the activity patterns in the homogeneous network and compare it with the activity patterns in the heterogeneous networks, in higher frequencies there was not much of a change. Only in the lower frequencies there was large change that was present. So the question he asked was, if I now suppress the low frequencies, would I be able to get back grid pattern activity even with heterogeneities manifesting in the structure? Right? So could the deleterious impact of heterogeneities on grid pattern firing be eliminated by suppressing low frequency neural activity? So there is a very a nice way of introducing suppression of lower frequencies in neurons. So there are specific ion channels which are slow, and they also act as negative feedback loops because say for instance this is a depolarization activated hyperpolarizing channel, or this is a hyperpolarization activated depolarizing channel, and therefore in effect they are implementing a negative feedback loop. And as I said, these are slow channels, which mean that they would be able to suppress only low frequency inputs and not high frequency inputs. So this is a chirp stimulus that is being presented to a neuron. Normally a neuron is an RC circuit, which means that you will have a low pass kind of characteristic. But when you have these kinds of ion channels present on top of it, what these will do is this will suppress the lower frequencies, the capacitance will suppress the higher frequencies, and therefore you will start getting resonance in these kind of structures. Right? So, and if you look at the entorhinal cortical neurons, they express Hc and Kv7 channels, and they resonate around the theta frequency range, which is 3 to 10 hertz basically. Right? So what Devyans did was he introduced this resonating system into individual neurons instead of taking a low pass characteristic, which was just based upon the time constant that we talked about. Instead of that, he introduced resonance through two different ways. One was through this high pass filter, which was just a differential that was introduced, and together they elicited resonance basically. This was more phenomenological, just to show that it doesn't matter how you get resonance, you will get suppression of lower frequencies. So you see that initial part of it, there is a huge suppression of lower frequencies, and you could also get that suppression with a mechanistic means similar to that ion channel that we talked about, where you suppress the lower frequencies again and you get resonance in the theta frequency range basically. So you introduced resonance into these, instead of having integrators as neurons, he made individual neurons as resonators, which were resonating in the theta frequency range. Lo and behold, in the network made of integrator structures, you saw that there was abolishment of the grid pattern firing with the highest degree of heterogeneity. And irrespective of how you introduced the resonance into individual neurons, either through the phenomenological manner or the mechanistic way, you get back this grid pattern firing in these particular structures. Right? So and this is not the only way to get it. There is another paper where they showed that continuous attractor networks with heterogeneities, there is a abolishment of grid pattern firing or pattern firing, and you could get that back by an inhibitory feedback loop. Instead of having an intrinsic structure which gives you that negative feedback loop, you just introduce an inhibition that also can give you stabilization basically. And so it turns out that the intrinsic properties of neurons, having them as resonators as opposed to integrators, can give you computational benefits which you never thought you could have with just integrators present over there. And here it doesn't matter. I mean, you could increase the level of heterogeneities and you still get stabilization over here basically. So that was a rate-based model. So he wanted to do this in biophysically realistic neurons as well. So instead of doing a 2D continuous attractor network, which is a donut-shaped structure, he just introduced, he took a much smaller, so the number of neurons here is around 3,600 or more. Different sizes were done over here. But here, it's just a ring network of 80 excitatory neurons and 20 inhibitory neurons. That's the I to I connection. There are no E to E connections. So you have that Mexican hat kind of connectivity for I to E and I to I. And he looked at propagation of activity along this particular structure. Right? I mean, what exactly would happen with time, and this is just taking the activity of all the I cells, taking the activity of all the E cells, looking at the firing rate, how exactly it, because it is propagation of activity, you will have repetition, and that is what is manifesting here in the reduced dimension as well. Right? So here again he introduced heterogeneities, but here, because these are biophysically realistic, you could perturb them much more, and you can have different kinds of heterogeneities present over here. So this is a homogeneous network, right? So in this case, what I said was this is a model for this kind of an ion channel basically, which introduces this. In a biophysical network, I don't need to model that. I can just get the, I mean, I will still model, but it is more biophysically realistic. I can take the exact same Hessian channel and ask questions about what would happen if I increase or decrease the Hessian channel density in that particular structure. And the control for that is, as I said, this is a slow negative feedback loop. If I don't want a slow negative feedback loop, which is what is going to suppress the lower frequencies, I just speed up the Hessian channel, a fast Hessian channel. And what you are observing over here is that, I mean, how many of the models is capable of giving me the patterned firing over there. And you see that with fast Hessian channel, there is no impact on the number of models that is giving you this patterned firing. But with the actual Hessian channels, you see that as you increase the Hessian channel density, there is an increase in the number of models that give you this patterned fire. Again, emphasizing the impact of that. At this stage, I mean, the excitability of the cell has become so low that you won't be able to fire, but I mean, within reasonable limits, you have that increase. So, this is for a homogeneous network. Now, if you introduce low degrees of heterogeneities, you see that there is a dramatic fall in the number of models. So here you had around 60, here you have 20 of them giving you this patterned firing. But again, here as well, if you introduce Hessian channels, you can increase the number of models that give you this pattern firing, and with higher degrees of heterogeneities, it almost falls to 0 with fast Hessian, and with actual Hessian, you can still get a fair proportion of models which can give you pattern firing, again emphasizing the importance of slow negative feedback loop in sustaining this kind of activity in continuous attractor network. This is currently in review in eLife. We are working on the revision basically. So in this case, the network heterogeneities hampered the performance of the continuous attractor network. And resonators which implemented slow negative feedback loops stabilized the heterogeneous continuous attractor networks that are present over here. Alright, so the third and the final part of this talk is recurrent networks. Anjana is here. And so she looked at different tasks. I'm going to be talking only about the simplest possible Go task. I mean, you have a recurrent network, those are the different inputs. So those are all additional, this is a one hot vector. So if it is one, that is that particular task. In this, it's the Go task. So there is a fixation stimulus, there is a stimulus that is fixation input, stimulus input, and when it changes the sign to response, it has to give you the output. This is the simplest possible Go task, and she has this recurrent network. Right? So she had a set of a complete framework for how to do this, right? On how exactly to study the impact of heterogeneities in neural circuits. So the first is that, I mean, you have in biological system, the learning occurs in the presence of heterogeneities, and therefore you can't just ignore that even during training. You can't just have homogeneous initialization. You should have a network which is initialized with heterogeneities and then train it basically. Right? So neural heterogeneities must be introduced into artificial network during training when the network is learning the specific task structure to achieve effective performance. That's one. Second, as we already saw, there are different forms of heterogeneities, and we don't know to what scale. So there are even suggestions that several pathological conditions are because of changes in the extent of heterogeneities, not necessarily because of the changes in baseline properties, but it's just that their heterogeneities either become higher or lower, and that is what is resulting in the pathological condition. So therefore, in this particular scenario, you want different forms of heterogeneities, intrinsic, synaptic, and other things must be introduced at different levels, not at only one particular level, because, I mean, if you just fix it at this, how do you know that it is that as opposed to something else? And therefore, the graded impact has to be assessed progressively rather than just at one particular heterogeneous versus homogeneous. And you can't just use single network for studying this, because, I mean, you have different networks have different connectivities, and the impact of heterogeneity on this network could be very different from what is there on this particular network. And therefore, you should have a population of networks with very different hyperparameters, and all of them should be assessed across all different levels of heterogeneity. Otherwise, you are going to be biased by the outcomes of this. You just study this with different levels of heterogeneity. That result doesn't necessarily hold with this. Right? I mean, I'll come to the results where that actually comes through. And then you have a scenario where even after training, let's say during the process of training, you have changed the neuronal properties or you have changed the synaptic weights, and that is what is giving you the outcomes. But in biological systems, it's not like you have trained that particular system and you're never going to touch it. There are going to be changes post facto. So you have that post facto training that is occurring, post facto changes that are occurring. So you will have to assess the impact of those post training heterogeneities or perturbations on the system, different kinds, different levels here as well. Otherwise, you are not really assessing what exactly is happening with reference to heterogeneity. So there is heterogeneity at the time of learning and heterogeneities after you have trained the particular system, basically. And finally, it can't be just one performance metric that you use for studying the impact of heterogeneities. You should have an array of metrics focusing on different aspects of network performance, and each of them should be studied together. All of them should be studied together, because I mean you could have two different metrics, and if you study only for one of them, you might be optimizing only one of those. On the other hand, there could be constraints on getting both of them together. There could be a front that is emerging, and you could have either high speed, low accuracy, high accuracy, low speed, things like that can come through, and therefore it's very important to study the performance of the networks, not just with one particular performance metric, but an array of techniques, basically. So this is the framework that she used. She did not just use one particular network. She used 53 networks in this case, each of them with 5 different levels of heterogeneities. In this case, intrinsic heterogeneities are shown over here. So that's the homogeneous network, and you have the time constants increasing across this particular different levels of heterogeneities. And you had a training system with the reward modulated Hebbian learning rule. So that's the task, and these were variable. It has to be variable because otherwise the network would just learn that particular pattern, not necessarily learning the task, and therefore you have to perturb the timing of the fixation and the stimulus periods and so on and so forth. And you compute the output, and you want the output to match with the go task requirements. And she will take the output during this response epoch and compare them, and she's going to ask questions about how exactly the network training and performance are dependent upon the different levels of heterogeneities that are present over there. So, and it turned out, so these are the different levels of heterogeneities. This is just the training performance, and that's the median. If you just look at the median, you will say that, oh, there is no impact of heterogeneities basically, but that's the beauty of biology. There is network to network variability, human to human variability, and that's exactly why what works in one human doesn't necessarily work on others. So you have this network to network variability. So if you look at network 33, you would, I mean, if you had just used this particular network, you would have concluded that, oh, the number of training trials is higher when you have heterogeneities as opposed to having no heterogeneities. If you looked at the blue network, which is N26, there you see that, oh, I mean, there is a red. If you have more heterogeneities, then the number of training trials is lesser. And if you look at the N39, you would say that, oh, there is no relationship whatsoever between heterogeneities and training performance. But I mean, that's the important aspect that her work brings out, that there is huge network to network variability in terms of how heterogeneities affect each and every network. It can't be something which can be just looked at from one particular network or just the median of the outcomes. Right? She projected the activity of these different networks. So that's the network number. H is the heterogeneity level. 0 is a homogeneous network. 5 is the highest level. So these are two different networks with heterogeneity 0, which means that there is no heterogeneity, it's a homogeneous network. So those are 4 different inputs, and she's just taking the activity patterns for those presentations and plotting them on the 3-dimensional reduced dimensional space. You see that the trajectory of how exactly it reaches its goals are very different. Even though the outputs are very similar, the network activity here is very different from here. And for the same network for 2 different heterogeneity levels, again the network trajectories are very, very different. When she takes this and uses canonical correlation analysis, project it onto a single structure over here and compares the trajectories, again there was significant variability. In certain cases, in certain networks, you would see that this is the distance of the network with H1 compared to H0 basically. And if you look at it, the distance here for H1 is lower compared to H5, so that is for network 35. So if you looked at only this network, you would see that if there are lower heterogeneities, then it would match with the homogeneous network, and if there are higher heterogeneities, it would be farther away. But if you come to N10, it is exactly the opposite. You would have H1 having the highest distance and H5 having a lower distance basically. So it's not necessarily something that is consistent across, and that is the need why that their framework came into picture. Different levels of heterogeneities, different hyperparameters, different initializations, and you have to have the heterogeneities during training, and that brings about this network to network variability basically. So it's important to account for all of this. And post-training perturbations, as I mentioned, so she looked at a bunch of them, again not just one particular level, different levels of perturbation for each of them, took all the networks, all the different levels of heterogeneities, introduced different kinds of perturbations over there, and the maximal impact was observed with synaptic jitter. And here also you would see that some of the cases the blue would be above the red, some other cases you would have the red will be above the blue, and so on and so forth. Right? So it's very important to account for this. You can't just take one network, one initialization, one level of heterogeneity, make a binary homogeneous versus heterogeneous comparison with one metric. The training performance was very different. The performance in terms of trajectory distances is very different, and so on and so forth. So it's extremely important to account for all of this. All right, so in this case, there is pronounced network-to-network variability in the impact of neural circuit heterogeneities on training performance, as well as activity dynamics of recurrent networks performing cognitive tasks. So this is just one task. She has an expanded version of this with different tasks, with memory, without memory, and looking at task dependence also, and the same network, same initialization, different tasks, very different outcomes as a function of heterogeneities basically. All right, so we have been working on heterogeneities for a long time. As I said, Poonam was the one who started this line of work in the laboratory, but since then we have been looking at different neuronal subtypes, different networks, different kinds of heterogeneities, what exactly their impact is on the function over the years. There are a bunch of review papers from the lab. This is with reference to stable continual learning. This is with reference to efficient information coding. This is the one that I said I will advertise. This came up recently, and even though we have, our names are very, very close to each other, we have not necessarily talked to each other, discussed each other. This paper has, as I said, our approach is to stay as close to biophysics as possible and have these heterogeneities introduced in a more biophysically relevant, biophysically realistic manner. This paper will give you very different ways of introducing heterogeneities from a mean field perspective. What happens to heterogeneities in a neuromorphic system? What happens when there are heterogeneities in oscillatory systems, and so on. So there are very different perspectives. We all gave talks at the Bernstein conference 2 years ago, 1 year ago, 1.3 years ago, and we came together to write this perspective, I mean, I strongly recommend this. Please read through this. It has heterogeneous viewpoints and heterogeneities and how exactly they impact neural circuits. Go through that. All right, so last few slides, if my, I mean, after this. Just to summarize this part of it, so we found that in decorrelating networks, both in terms of channel decorrelation and pattern decorrelation, we found that heterogeneities had a beneficial impact on performance and robustness. And it hampered performance in continuous attractor networks, and that was restorable with the introduction of slow negative feedback loops, in this case with ion channels, but you could do it with inhibition as well. Right? And there was network variability in the impact of heterogeneities on training and dynamics of recurrent networks performing cognitive tasks. And this is something which came up from several different lines of work from our own lab. So the diversity that we are observing, in one case it is beneficial, in another case it is hampering, and in a third case there was huge network-to-network variability. So that diversity depended upon several different factors: the kind of network, the type of model used, the network hyperparameters, the task being performed. When we have lines of evidence from Anjana's work for this, type of heterogeneity, the degree of heterogeneity, the method to introduce heterogeneity. As I said, there are different ways of doing things. The metrics used to evaluate performance and interactions between the different forms of heterogeneity, all of them play a role, which is why we have that 5-point framework for how to assess the impact of heterogeneities on neural circuits. So now if this is all so complicated, how do we reconcile this diversity of impact of heterogeneities on different networks? How to even approach the problem of heterogeneities and neural circuit? Is there an overarching framework to study this? So in all of neuroscience, we have been kind of asking this question. At one end of it, we have one-to-one relationship between structure and function, right? Where there is that discredited idea of phrenology. And at the other end of it, we have a scenario where everything contributes to everything else, and there is no real structure associated with it. Right? So bringing that same notion to the idea of heterogeneities. So here, so far, people who have been looking at heterogeneities have been looking at specific forms of heterogeneity, specific degrees of heterogeneity, specific networks, and have been showing that, okay, there are specific forms of heterogeneity that yield robustness and resilience to a network that we are looking at. And at the other end of it, you could have a scenario where, I mean, it doesn't matter what kind of heterogeneities are there, what kind of components they are introduced, and they will give you better resilience, better robustness, basically. So that's one end of the one kind of, I mean, whitewashing the whole thing and saying that heterogeneities are good, we should have it, otherwise it's not going to work. But I mean, our own work has shown you that in some cases it is hampering, in some other cases it is beneficial. So that kind of whitewashing is not something which is possible in this kind of structure, basically. So, and whenever the question in neuroscience has been X versus Y, the answer has always been X and Y, one way or the other. And in this case, also the answer is somewhere in the middle, but there is a name for what exactly that intermediary is. It turns out that, I mean, it could be completely well understood, or I mean, at least from a conceptual framework, you could have a complex system, or to be more precise, a complex adaptive system, which are neither fully determined, where one specific form of heterogeneity will give you robustness or resilience, or you have a completely randomized system where you can have arbitrary heterogeneities in arbitrary components giving you your collective goals. So complex systems are defined by their intermediate levels of randomness. That's where the complexity is high. And we have been looking at the complex adaptive system from single neuron perspective, from a network perspective, and so on and so forth. I didn't talk about it today. One of the prominent lines of work in the lab is on degeneracy, basically. So the complex system framework provides an ideal theoretical framework for studying heterogeneities and its impact. I would also strongly recommend this paper to all of you. You are neuromorphic scientists, but neuro part of it is a biological system. Therefore, I would strongly recommend this. I come from an engineering background. I have a PhD in electrical engineering, and then I moved over to biology. This paper changed the way I look at biology. Right? So this is by Gerald Edelman. He got his Nobel Prize in Physiology or Medicine for antibiotic structure in 1972. Then he saw light at the end of the tunnel, switched over to neuroscience, and had some important papers and books written on it. Some parts of it is controversial, but this is a landmark review basically. Right? So in that review, there is this definition, quote unquote definition of a complex system. It's extremely difficult to define a complex system. A complex system may be considered as one in which smaller parts are functionally segregated, or we'll come to this in a minute, or differentiated across a diversity of function. Just remember that there are functionally segregated smaller parts, but also as the one that shows increasing degrees of integration when more and more parts of it interact basically. Right? In other words, a complex system may be viewed as one that reveals an interplay between functional specialization and functional integration. Right? I mean, you could put this idea together. You could say that there are functionally specialized subsystems. So this could be at the molecular level, you could have them as different ion channels, or at the cellular level, these could be different cells. This could be an astrocyte, this could be a pyramidal neuron, this could be an interneuron, and so on and so forth. And when they interact, not all-to-all interactions, not random interactions, but specific motive-based interactions over here, if you have that kind of an interaction, that is the functional integration part of it, that gives you collective functional outcomes. So in this case, what I am interpreting with reference to the idea of heterogeneities is that these are heterogeneities in individual components, not just, binary in terms of whether they are present or not, also the degree to which they are present. And it's not one single heterogeneity that is going to give you functional resilience or, better performance. You should have different heterogeneous subsystems which interact with each other in specific manner, and that is going to be system dependent, that is going to be network dependent, that is going to be task dependent, and those interactions between the different heterogeneities, specific kinds of interactions, is what gives you robust neural circuits. It's not arbitrary heterogeneities. You can't just put arbitrary heterogeneities in a network and expect functional outcomes to be robust or better. It has to be specific kinds of heterogeneities, and one kind of heterogeneity, one degree of heterogeneity should be dependent upon the other kind and the other degree that is present. Then and only then you will have functional outcomes as you expect basically. So that's the framework. As I said, we have been projecting everything to the complex system framework from our laboratory. It's one of our favorite pastimes, right? So this is, to us it feels like an important step forward, and you can bring in all of self-organization and other things into this into this framework without loss of generality. All right, so to finally conclude, so neurons are not homogeneous repeating units. Heterogeneities are extremely important, and this is the same thing that I mentioned. The network performance was either hampered or improved in different networks based upon several set of factors which include these, and do not make generalizations about the impact of heterogeneities based on just one choice of this. Don't just take one kind of network, one type of heterogeneity, one or where there is no interactions between different forms of heterogeneity, and say that heterogeneity improves function or heterogeneities hamper function. That would be non-ideal, that would be wrong. The global structure of different heterogeneities, that's the important keyword. It's the global structure that is important. It's not just individual components. Global structures of different heterogeneities and how they interact with each other are extremely important. A complex system perspective, we think, can offer resolution to how different forms of heterogeneities can alter network performance. Thank you so much.
there is any